So, we will plan it this way, we will move on now, move on to the physiology of digestion, and you must have noticed that whereas we have made an attempt at understanding the skeletal muscle system and then of course, the cardiac muscle system, but third another equally important component, that is the smooth muscle system. So, this is our opportunity to take a look at the smooth muscles, because they form an integral, the digestive system is driven by the smooth muscles, not only the digestive system, but a range of other systems, and let us dive deep into it and try to find out what. Okay, so alimentary canal, movement of food through gut, secretion of digestive juices, absorption of water, circulation of blood, control functions, we will do all this in due course. So, I will ask you something, if you take a transverse section of human body somewhere here at the level of the waist, okay, just take a section and look at it, okay, and you will get in a diagrammatic, if I were to draw a diagram, it will look something like that. Look at the top there, you see the top, that is the spinal cord, that symbolizes the spinal cord, okay, just see spinal cord. Below that there is a slightly not so dark circle, below that there are 3 circles there in a row, and then there is space below the 3 circles. The topmost there is the spinal cord, below that is the centrum which represents vertebral column, and below that you have, can you see that hollow circle, tiny circle, that represents your dorsal aorta. This is a plan true for all vertebrates. Okay, take a section, you will find that there is what? Spinal cord, which is in a vertebral column, below that there is dorsal aorta, which is taking the blood backwards from the earth backwards, and below that if you see there is this huge cavity which we call as peritoneal cavity, and if you look at that I can say that there is a big tube and inside that there is another tube. So, basically all vertebrates have what you call as a tube in a tube organization. Are you getting my argument? Outer tube is your body wall and inner tube is your alimentary canal, and it is because of a tube in a tube structure, appreciate the point. Therefore, it is possible to have the inner tube that goes in and out, in and out, in and out, in and out, and therefore, it is possible for you to have an intestine which is 20-25 feet long. Are you getting my argument? It is only that is possible, and we need that length of intestine because we need so much room, so much space to facilitate digestion. So, you need we have a very long alimentary canal, and we always wonder how is that alimentary canal accommodated in our system. It is because this is a tube in a tube. Okay, the inner tube can go large number, but now this space that you have between the 2 tubes is called as peritoneal cavity. What do you call it as? Peritoneal cavity. What do you call it as? Peritoneal cavity, and all vertebrates have what you call this peritoneal cavity. Good. Now, you might have heard, you know, they when they give injection or administer into the drug, inject drugs into the body, often you might have heard intraperitoneal route. Intraperitoneal route. Why? What would I mean by IV route? Intravenous, very good, intravenous route, very good. So, there is another blood which more common is intraperitoneal. So, intraperitoneal route you essentially introduce your noodle in this space and you give there is plenty of room there, so you can give a large volume intraperitoneal. Okay. And then, but you see, remember, if this is the alimentary canal, this symbolizes alimentary canal with this duodenum, whether it is duodenum or whether it is ileum or intestinal large intestine, this is signified there. It is connected to the body, rest of the body by means of this membrane which we call as mesentery. So, the stomach and the intestine, okay, and all that is ultimately supported not anywhere, no, it is connected to the rest of the body by what? Mesentery. So, where is the mesentery coming from? The mesentery comes from the dorsal side, it comes from the dorsal side. Are you with me? And that mesentery is important because the blood supply to the entire alimentary canal, which is, you can say, the arteries going there, the blood vessels which will bring the deoxygenated blood back to the heart, and also the nerve supply. Everything is through the mesentery. So, just look at this diagram. This is your alimentary canal. It does not have anything yet. So, everything is through what? Through mesentery, and that is very well shown in. Okay. But before that, Let us also take a look at the different parts of the different layers. Do you remember we have seen in the histological section the outermost layer was called as serosa, which was a thin layer. Inside that, what was the layer just below the serosa?
Muscularis.
Ah, very good. Longitudinal muscles. What do you call them as? Longitudinal muscles. So, here you have a layer of what? Serosa, longitudinal muscles. Longitudinal muscles go along those muscles whose length is organized along the length of the entire alimentary canal. What will you call them as? Hello. When I say longitudinal muscles, oh, your esophagus goes this way. Okay? It goes this way. So, those cells whose long axis is parallel to the axis of the esophagus will cause longitudinal muscles, and those which go round will call as circular muscles. So, there is outermost layer of longitudinal muscles, then there is a layer of circular muscles, okay, and then there is a layer called as submucosa, and then there is the mucosa. Okay, okay, good. But life is more interesting than that. Listen to this. What was the outermost layer I said? Longitudinal muscle layer. Very simple. What did I say? Follow the point, okay, the things are going to become interesting. Outermost layer is longitudinal, outermost layer is serosa, which is not muscle. Inside that is it's like epithelial tissue, thick epithelial tissue, tough, which protects alimentary canal. Inside that is a layer of longitudinal muscle layer, which is pretty thick, where it plays a very important role in peristalsis. All these muscles they are going to, and then inside that is circular, a layer of circular muscles. In between the layer of longitudinal muscles and the circular muscles, there is a very interesting layer of neurons. What did I say? Neurons. And there are millions of them, okay, and it is called as mesenteric plexus. What do you call them as? Mesenteric. My refers to muscles, enteric refers to alimentary canal, okay, a layer that is there in the muscles of the alimentary canal. What do you call it as? Mesenteric plexus. What do you call it? You have to, this should fit here. What did I tell you just now? Mind, it's millions of neurons, okay. And then there is circular muscles, okay, and then inside circular muscles there is another layer again of neurons, which is called as submucosal plexus. So, again serosa, longitudinal muscle layer, circular muscle layer, in between, can you see this black line with those round dots? What do you call it as? And inside that is another, what do you call it as? Submucosal plexus. These are the entire game of the alimentary canal with reference to peristalsis, when to begin, how to begin, when to terminate, everything is controlled by these neurons. Are you with me? So, we have been knowing for a very long time that peristalsis happens. Who controls the peristalsis? Mesenteric plexus and the submucosal plexus, and these two plexuses in turn get innervation from the autonomous nervous system. So, the brain, autonomous nervous system, sympathetic, parasympathetic, they come and talk to the mesenteric neurons of the mesenteric plexus and the submucosal plexus, and overall they will control the
Peristalsis.
Why only peristalsis? The secretions, very good, secretions, and well, you have been starving, so reduce the blood supply to the alimentary canal. You have just had food, increase the blood supply to the alimentary canal. Are you with me? Okay, this all happens without our knowledge. Okay, again, these plexuses have a very important role in all those actions. Not only the smooth muscles, also the smooth muscles which line the blood vessels of the alimentary canal, so that you can control the blood supply when you want it, and when you do not want it, you stop it. And this is this of course is the mucosal layer. Oh, this is interesting. It is the same thing as the original one, but you can see the mesentery there. Through the mesenteries, you can see the red one which is taking the oxygenated blood there. You can see the blue one which is taking the deoxygenated blood there, and that yellow one here. Outer one is the mesenteric plexus. The inner one is the submucosal plexus, and then you can see the different layers. The inner one most is the epithelial, then the mucosa. Then you can see the different glands. Spend some time on this. Understanding this image, you will appreciate what it is. Okay. Now, we have again you know what is at the top, skeletal muscle, you know what is at the bottom. What we are going to try to understand right now is what muscle there. So, let us focus on smooth muscle. Okay. Now, before I start smooth muscles, it is a different world. Smooth muscle is a different world. Okay, it's neither cardiac nor it's completely different. The only thing that is common is there is an actin filament, there is a myosin filament, and they two slide on one another. Other than that, everything is different. Hammer the point. It's different. Okay, don't carry any baggage from the skeletal system or cardiac system. No, it's a different system, and we need to spend time on understand and to understand how it is organized and how it is controlled. Oh, just a nice image, the electron photomicrograph showing the top one you can immediately understand. Each is a single cell. Are you with me? The muscle fiber, and of course, you do not see inside there will be one nucleus here, one nucleus there. Okay, you will have cells there, but smooth muscle, just how it looks, how different it is. There are bundles and bundles, and they go all over. You can't find a pattern, there is hardly any geometry there. Look at the geometry there is hardly any geometry. Okay. But if you go deep into this and if you try to look how a single cell looks like, the single cell looks something like this. This typical shape is called as spindle shape. You know how this word came in, this word will come very often in biology, and I will tell you how this word came from. You know, in the textile industry, you make cloth from the thread. Okay. The thread, the axis on which the entire thread goes, okay, which you also put in the receiving machine. Okay. So that would look it's called a spindle because it looks like there is more thread in the middle, less. So it's thick in the middle and tapering towards the end. So the word has come spindle shaped. The word we have borrowed from textile industry. So there is a spindle shaped cell. Do you appreciate now? Goes on tapering to the ends and thick in the middle. What do we call that? Spindle. But you would wonder as to why that word spindle is coming from, therefore is spindle shape. Okay. So there are thousands of spindle shaped cells come together, and perhaps some of you may appreciate that the slide that we saw of the smooth muscles very much looked like this, and in a single cell there is a single nucleus. Okay, not too many nuclei in a single as we have in the skeletal muscles, and this gives you the dimension. What are the dimensions? About 2 to 10 microns in diameter and about 100 to 200 microns in length, but depends on the organ. Most not all. Smooth muscles show another phenomena with which again we are familiar, and the phenomena is the occurrence of gap junction. Can you see the gap junction there? Hello, can you see there in the top? A neuron, I made a mistake again, not a neuron, a muscle cell, a smooth muscle cell, okay, is talking to another muscle cell by way of gap junction, okay, and through which of course the ions can pass, something like our intercalated, okay, and the proteins here are called as annexins, okay. So you have, so you have one cell and connected by gap junction. If I focus here and if I draw a diagram, I will send one cell, and if one cell is excited means suddenly calcium ions have gone up. That principle still holds. What is the principle? If you want actin myosin filament to slide on one another, you will have to suddenly give a flush of what? Calcium ions. Okay, and if that happens within one cell, the calcium ions could pass from one cell to another cell, and other cell could also now that calcium ions, it could also respond. Okay, and it could, and then it is supplied by, and then at the top of that you can see a neuron. Can you see a neuron there? Okay, and this innervation of a smooth muscle is anatomically slightly different from that of skeletal muscle. I will talk about it very briefly. So if this neuron is a part of the postganglionic parasympathetic, what neurotransmitter is being released by those terminals there that you can see? Very good. What neurotransmitter? Acetylcholine. So if acetylcholine comes here, okay, then if this cell is stimulated, it may stimulate the next cell. Okay. So you do not have to, every cell need not be equipped with its own nerve supply. This does not sound very great, but it is true for a skeletal muscle. In the case of skeletal muscle, every muscle cell must have a fiber which is coming, okay, from the spinal cord or brain or whatever, and release what neurotransmitter there? Again acetylcholine. Again what? Acetylcholine. I am sure you can appreciate here I have one smooth muscle cell, another smooth muscle cell, and author tries to draw our attention with the help of using an image of electron photomicrograph. What is author trying to show us here? Gap junction. What are you seeing here? It is gap junction. Okay. So, let us anatomically find out what those gap junctions are there. This we have done 100 times. Hello, are we there? Where are we? Spinal cord. Okay. And this is your skin, and you touch there or pick a pin there, pick up. Yeah. Am I said something right or wrong? Prick a pin. Okay. And then you excite the pain receptors, and then as a result of the action potential will start. Okay. So, there is a transducer. Okay. Any perturbation here will start the action potential. The action potential go via the dorsal root ganglion. It will enter into the spinal cord via the dorsal root. The processing of information will happen via the interneurons, and this particular neuron, the blue one there, the cell body is located in the spinal cord, is what is a cholinergic neuron that will give rise to a fiber that will come out from the ventral root. It will go all the way and supply the skeletal muscle, and the neurotransmitter that is being released is again acetylcholine and is acting by a what receptors? Very good. Say that again. Nicotinic. What receptor? Nicotinic. And this is extremely monotonous, very standard, whether you are a fish or a bird or a reptile. This is it when it comes to innervation. Now, this is so different from the smooth muscle, because in smooth muscle it can be excited by a neurotransmitter, it can be excited by a neuron, it can be excited by tissue hormone, by an autocoid, or just by stretching it. It's enormous diversity is there when it comes to stimulation of what? Smooth muscle. It's extremely diverse. Okay, I will talk about it. This is skeletal. This is most important. Now, let us compare it with the scenario in the of the innervation of an organ, okay, which is equipped with smooth muscles. Now, smooth muscles, one very simple definition of the where you will find smooth muscle is you will find it in the wall of a hollow organ. What did I say? Wall of a hollow organ. Well, stomach is hollow. Am I right there? Duodenum, hello. Intestine, enter. It's hollow. Okay. Gallbladder, hollow. Bile duct, hollow. Pancreatic duct, hollow. Urinary bladder, hollow. Uterus, hollow. Okay. Dorsal aorta, hollow. All these organs are equipped with what muscles? So, one definition of smooth muscle is whereas the skeletal muscles will anchor themselves on a bone, the smooth muscles will line the hollow organ. Is the definition there? And here is a classic example of a hollow organ, in this particular case a part of the alimentary canal. Does it have, just as our skin has sensory function, do the visceral organs have a sensory function? What do you guess? What do you think? Yeah, why? Yeah.
For example, if food is passing, it has a sense that the food is passing, and then it has to perform peristalsis in response.
You don't know about it. Why should you know about it? Why should you? Yeah, you are right, but you don't know. Why do you need a sensation for that? You don't need. You don't tell the angle gland to secrete the enzyme. The gland secretes on its own. What do you mean by that? It's out of your control. You have what control do you have? No, it's autonomous nervous system. When the food arrives, okay, into the duodenum, okay, the wall of the duodenum knows that the food has arrived. It will send an endocrine message to the gallbladder. The gallbladder will squeeze and pump the bile out, which will pour into the duodenum and do its business, whatever it is. You have no role.
Is it the case that if in any abnormal condition we'll get to know about it? Like
you got it, he has given the correct. If it stomach pain, gentlemen and ladies, pain is there, okay, something goes wrong. When something goes wrong, the thing that has gone wrong wants to tell your brain, okay, that something is not quite what it should be, the pain, okay, and stretch, okay, and discomfort. You can use all those words. So, any such phenomena that is happening here, it takes information again by the dorsal root again to the spinal cord. So, you have this system also in the spinal cord, and here it is processed, and now this is actually a part of what? Autonomous nervous system. Autonomous nervous system, it goes to the ganglia. Are you with me? In this particular case, sympathetic ganglia, okay, the preganglionic fiber is going to have what neurotransmitter? The preganglionic fiber is going to have what neurotransmitter even very good, and this is since this is sympathetic ganglia, the part of sympathetic nervous system. So, this blue guy is going to have what neurotransmitter? Very good. What neurotransmitter? Not. And having entered here, the terminal here, okay, it will, this may either directly talk to the smooth muscles or it may talk to the mesenteric plexus neurons. Are you getting the story? Both the possibilities are there. We will talk about it later. It can talk to what? Mesenteric neurons. So, and then mesenteric neurons, they take over the charge because they are sitting right in the alimentary canal. And okay, so this was the point I have already made. Here this is the typical skeletal system, and there is a nerve fiber that is coming from the spinal cord. They are all cholinergic, and each, you see, this axon gives rise to one branch, second branch, third branch, fourth branch, as many, and at each junction it is releasing acetylcholine. Look, if I cut here, then this muscle cell will not get acetylcholine, and if it does not get, this won't contract. Very simple. Every guy has to have its own supply of acetylcholine. Every muscle cell skeletal has to have. Not so here. Here we have already seen there is a nerve supply, and these are what you call as the beaded extensions. They will release their neurotransmitter, but if one cell is excited, gap junction, second cell, third cell. So, it will continue. So, what are the smooth muscles? They primarily support visceral function. Visceral means in your alimentary canal. If you open the body wall, whatever you have, you have liver there, you have pancreas there, you have different parts of the alimentary canal. All those organs we will call as what? Visceral organs, and then each fiber is individual small cell. They lack okay, yeah. Another interesting point. Whereas the sarcomere, you remember again skeletal muscle system, I will say it is the heart and soul of the skeletal muscle. Okay, if you have understood sarcomere, it's sarcomere, okay, because one sarcomere, 100 sarcomeres, they are all same. We have to understand sarcomere, how the actin filaments are inserted and how in between thick and thin sarcomere. In the smooth muscle, there are no sarcomeres, okay, and that beautiful geometry that you get in the skeletal muscle, sarcomere, and then perfect distance, okay, all that is not there. No, that's not there. Then how does it work? I will show you that organization in the next slide. Here we are looking at one complete smooth muscle cell, diagrammatic, and it is talking to this cell by way of gap junctions. It is also talking to this cell by gap junctions, and you can, the arrows indicate the influx and outflux of the ions, so that the messages can go. Spend some time on what? On a single smooth muscle cell. Look at the smooth muscle cell. First of all, I will draw your attention to the plasma membrane, and you will find that as you go along the plasma membrane, there is a certain depression. You get that depression here? Those depressions are called as, a single is called as caveolus, plural is called as caveoli. It is just a depression in the plasma membrane, but it is a matter of great attraction for the physiologists, because whenever this cell receives any information from anywhere, be it from the neuron, be it from a hormone, be it from a local hormone, any information anywhere, invariably the receptors which are sitting on the smooth muscle cells are located in this part of the caveoli. Those proteins, you know, which will get the message. Okay, means what? Okay, norepinephrine, action of norepinephrine or action of acetylcholine. How? By way of muscarinic receptor. Okay, muscarinic. Where is that muscarinic receptor? Most probably it is sitting here. It is in the cavity. What am I trying to do? I am trying to draw your attention anatomical peculiarity, okay, and I am trying to emphasize its physiological importance. Why is cavoli important? Because it is the site for accommodating a large number of receptors through which a signal will be received by the cell. Second thing I notice, just next to the caveoli there is this deep red patch, which are very weakly developed as compared to skeletal muscle. Sarcoplasmic reticulum. Is it there? Yeah, it is there. Sometimes it is there, sometimes it is not there, and when it is there, it is very weakly developed. Okay. Why is it so weakly developed as compared to the, what does it contain? Same, calcium. it contains calcium. Okay. Why isn't as extensively developed? It's not extensive, because these cells are relatively small, you know, and because they are small, you can always get calcium from outside. Okay. So, the nature has thought there is no great point in providing a great source for calcium within the cell, you can always get it from outside. Okay. Not so in the skeletal muscle. Okay. It is so deep inside, okay, and there you need a burst. Okay. So, therefore, those cells have an extensively developed sarcoplasmic reticulum, which is an immediate source for supply of huge amount of, not really necessary here. Okay, good. Second thing that I want to draw your attention is to these dark blue bodies which are seen in several places. You will call them as dense bodies. What do you call them as? Say that again. A dozen or more, I do not know, much more, I am sure much more than dozens, different proteins. Okay. Different proteins, they come together and this formed. Whenever it is, it may be sitting right in the cytoplasm, or it may be attached to the plasma membrane. If it is attached to the plasma membrane, then we call it as attachment plaque. It is the same thing. Dense body, if it is sitting on the plasma membrane, we will call it as what? Attachment plaque. Its very important purpose is to anchor the actin filaments. So, these are the actin filaments, these are the actin filaments. So, if I were still use compare which cannot be compared, it is something like Z membrane. You remember Z membrane? What was the purpose of Z membrane there? To allow what? Actin filaments to anchor, okay, and in a perfectly geometrical, there is no great geometry here. Okay, but it serves the same function that it allows. And if on this dense body there is I can see here the 3 actin filaments, there are hundreds, and here also there are obviously those red rods. What do they represent? They are myosin filaments. Okay, and the function is same. The myosin filaments will pull the actin filaments. When it is pulled, then of course these two dense bodies will be pulled. Okay, and that's how the cell will undergo its, okay, and then of course there are mitochondria all over. Here is a 3D image. The first one is a smooth muscle relaxed, the another is I just want you to compare this. This is a dense body, that is a dense body. Over that you can see the thin fibers which are the actin filaments, and right in the middle there are dark myosin filaments. Can you see there? It is very clear, we have done it, actin myosin filaments, and as a result of contraction, whenever it contracts, or rather it contracts because it slides. There is more of, and as a result of sliding, can you see in this image there is more of sliding here? You just compare that with that, and as a result of the entire length of the muscle is reduced. Okay. So it is, so you can compare the in 3D you can compare the smooth muscle cell almost in action. Okay.